Nucleic acids are a particularly important biological molecule, not because the others are not important. Carbohydrates, lipids, and proteins absolutely have incredibly important functions in living organisms. And a functional, healthy cell is going to require all of those biological organisms. But nucleic acids are particularly important because they are responsible for containing our genetic information. Nucleic acids store our genetic information, express our genetic information, and pass it on to the next generation. So our DNA, our nucleic acids, are important in passing on information to the next generation of cells and also for providing the instructions for cells to function, to produce the molecules and products that they need to survive and be successful. There are two types of nucleic acids. Deoxyribonucleic acid, or DNA, is responsible for storing genetic material or genetic information, which is encoded in a sequence of monomers. So basically, DNA speaks in a code that our cells can read and interpret to produce products for our cells. Generally, those products are going to be proteins. We also have ribonucleic acid, or RNA. RNA is important because it decodes DNA into instructions to link together amino acids. In other words, RNA is the interpreter that takes the information from DNA and uses it to construct proteins. The monomers of our nucleic acids are called nucleotides. There are only 5 different nucleotides. Unlike amino acids, where we have 20 different options for our nucleotides, we only have 5 options for monomers in our nucleic acids. Each of those nucleotide monomers is going to be made up of 3 parts. One is a phosphate group. A phosphate group is a phosphate linked to 4 oxygens. Our monomers also contain a 5-carbon sugar, either ribose for RNA or deoxyribose for DNA. That ribose sugar is attached to a 5-carbon, that ribose sugar is a 5-carbon sugar that only differs between DNA and RNA by the presence of a hydroxyl group at one corner. Deoxyribose does not have that hydroxyl group. Ribose, on the other hand, does have that hydroxyl group there. So this image up here is going to be your DNA. This one down here will be your RNA. The last component is a nitrogen containing base. So it's a base because it can accept hydrogen ions. It is, if you recall, our amine group is a nitrogen that is willing to pick up hydrogen ions. So that's why our nucleotide nitrogen region is called a base. The nitrogen region is made up of a nitrogen ring or of 2 nitrogen rings. So here I've drawn only one. This molecule up here is an example that has 2. Instead of carbon at all of its points, it actually has nitrogen at some of them. So that's where we get our nitrogen containing ring. So our monomers are made up of a sugar, a phosphate, and a nitrogenous base. This whole structure is one monomer, one nucleotide that is going to be linked together to other nucleotides to make our nucleic acids. Our nucleotides are linked together into long polymers that can be millions of base pairs long. And when they are linked together by dehydration reaction, we get a long chain connected phosphate to sugar, with the nitrogenous base in the center. The overall structure of DNA is 2 strands with the bases coming off of the center. Bases coming off of those strands. They're going to be connected in the center by the base. So inside we have our bases. Outside we have our phosphate and our sugar. Those strands run in opposite directions. That's going to be important when we learn about DNA replication and gene expression. DNA contains deoxyribose, and in the center, our nitrogenous bases are adenine and thymine, which are always found complementing each other. In other words, an adenine in the center will always pair with a thymine. Our other 2 bases are cytosine and guanine. Now, cytosine and guanine are always going to pair with one another at the center of our double helix. Now this picture is a little bit off because it's showing all kinds of different colors in there bound together. You're always going to have adenine bound to a thymine and a guanine bound to a cytosine. When you get your 2 backbones together, they twist to form this double helix structure. It's basically if you were to take a ladder and twist it on either end in opposite directions, this is the structure that you would get. This is sometimes called a twisted ladder structure. So a single DNA strand is made up of multiple monomers. Remember that our nucleotide monomer is a phosphate, a sugar, and a nitrogenous base linked together. We form a dehydration reaction that is going to link our sugar to a phosphate group. And that is just going to be a repeating pattern all the way down. Sugar to phosphate to sugar to phosphate to sugar. Sticking out of the side of the strand is going to be our nitrogenous bases. And that single strand with the sugar and phosphate backbone is going to come together and interact with another strand that is also a sugar phosphate backbone with the bases in the center. Those 2 strands are 2 DNA strands are held together by hydrogen bonds or hydrogen bonding. Now this is important later when we get to the function of DNA for DNA replication and for gene expression, because these hydrogen bonds, because we can pull them apart, we can actually separate our DNA strands, and that's going to be important later when we need to access that information for replication or gene expression. So our 2 strands come together and form this helical structure. RNA is really similar to DNA. RNA forms a backbone of sugar and phosphates, just like DNA does. However, RNA is single-stranded. So RNA is made up of only one sugar phosphate backbone with nitrogenous bases coming off of it. RNA is always going to be single-stranded, but it does not form a helix. RNA can actually fold in on itself, similar to what we saw in proteins. RNA can fold in on it to itself into specific structures that are, again, related to their function. There are several forms of RNA, all with slightly different structures. All are single-stranded, but they're all folded a little bit differently in order to perform the function that they need to perform. We will come back to the different forms of RNA in later chapters. A couple other key features of RNA. RNA uses ribose. This image here is actually DNA, so I'm going to draw the hydroxyl group on that. Again, phosphate, sugar, nitrogenous base, but the sugar is ribose with that hydroxyl group there. Another difference, is that RNA contains a fifth nitrogenous base called uracil. Now, if you remember from DNA, contained 4 bases: adenine, thymine, guanine, and cytosine. In RNA, we do not have thymine. So there's no thymine in RNA. Instead, we use the base uracil. It's a similar structure, but it's a slightly different molecule. So 3 main differences between RNA and DNA. One is that RNA is single-stranded. RNA contains ribose instead of deoxyribose, and RNA contains uracil instead of thymine.